Hello, this is Dave Tabb, and today I would like to talk to you about how to do deconvolution. I've been doing a lot of courses, as you know, on how to do top-down identification, and the topic of deconvolution is certainly one that shows up almost every time top-down identification is mentioned. So today I'd like to show you the Thermo FreeStyle 1.8 software. It comes along with XCalibur 4.5, at least FreeStyle 1.8 does, and then we've upgraded the FreeStyle software itself to SP2. Both of these are available from Thermo FlexNet. Now I'm going to be using a pair of raw files. You can see that their names are pretty long and ungainly, but they appear on this slide. These are both drawn from PXD 032724. This is a set we've recently published with Hilde de Roos's team here at Institut Pasteur. The data were collected, as you can see, back in 2014, so they're relatively compact and easy to process, and I think that they'll be good examples for us to walk through. Okay, so let us go ahead and start with kind of our script for how things are going to work out today. I'm going to start with the PXD number again. This is Proteome Exchange, so you can download these two files named here directly from Proteome Exchange. They were uploaded via Massive, so you can grab them from Pride or you can grab them from Massive itself. And I want to stress that these two files represent very different kinds of experiments. The top one is a data-dependent acquisition, which is to say that it is looking at which ions are most intense in the MS scan and attempting to preferentially produce tandem mass spectra of precursors that have intense signals in the MS scan. So you don't need to know in advance which proteoforms are which in this case. You simply need to say, I want to grab tandem mass spectra of as many high-quality looking precursors as I can. So the data-dependent acquisition experiment is very similar to what we think of as shotgun experiments in bottom-up proteomics, except that the ions we're sampling here are not short peptides, but rather intact proteoforms, at least hopefully. So the data-dependent acquisition experiment is one that we'll be looking at a little later in the presentation. But for now, I would highlight this experiment, which is an inclusion list experiment. It's a very different kind of experiment because here we know in advance which mass to charges are likely to have ions that are interesting to us, which ones are most likely to represent proteoforms that we want to measure. So this experiment is not simply a grab a tandem mass spectrum of whatever you can. Instead, it is focused on getting high-quality tandem mass spectra of these particular enumerated precursors. Now, it's very nice that this experiment was done, but I arrived at the lab after the postdoc who had collected this experiment had left. As a result, I don't have the spreadsheet he used to specify which particular precursor ions are most interesting. So for this, I'm going to make use of ProteoWizard. Now, a lot of people think about ProteoWizard as something you use to make an MZML file, but ProteoWizard has a lot of other useful tools built into it as well. And today, we're going to look at one that is very evocatively named Thermo Raw Meta Dump. It's a very exciting name, and we're going to use that to dump out methods from these two experiments. Now, if you're accustomed to using the MS-Convert GUI, you might think, gosh, I'm grateful that all these tools have graphical user interfaces. But in fact, Thermo Raw Meta Dump doesn't. We're going to have to run it on the command line. So in this case, I've already opened my command prompt right here, and you can see that I have already navigated to the directory that contains these raw files. I did my cd, c research, blah, 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 to the directory that contains my raw files. I did a quick directory on this location to show me the files that are currently in here. You can see that I have this raw file and that raw file. I note I have already performed a deconvolution of the shotgun experiment. I did that because it takes too long for me to wait for it in this video. So I've pre-done this one. We'll go through that process again, though, for the second raw file. But remember that we're not ready to do our deconvolution yet because I want to learn something about what precursor ions were specified for assessment in these experiments. Now for this, I need to tell the software where to find ProteoWizard. And I've installed it inside program files. I hit Tab to do autocomplete there. type PRO, I hit tab, and it says, there's a subdirectory that starts with PRO. It's called ProteoWizard. Inside ProteoWizard, I have another directory with the specific version number because some computers have multiple versions of ProteoWizard installed. And then inside that, I want to call Thermo Raw, and I can't remember the rest of the name, so I hit tab, Thermo Raw Meta Dump exe. Now, if I run that with no parameters on it, you can see that it gives me a very, very short description of how it works. In this case, I don't really need to do anything very special. I'm just going to run the software itself. I'm going to provide the name of the raw file. I highlighted that and hit enter to copy it to the clipboard, and then right-clicked to paste the raw file name there. And I don't want it to just spit all that text out to the screen. Instead, I want to capture that in a text file. And I'm going to call that one the same as the raw file name, but with CFG thrown onto the end. This is the configuration that we use to create this raw file. Now I'm going to repeat that command, but this time you can see it takes very little time. It's much faster than an MS convert because it doesn't need to look at all the spectra. It just needs to look at the methods for that experiment. So now I'm going to run that on the second raw file. And again, I want to output the configuration information to a file. And now when I do a directory, I see that I have this file.config and that file.config. These are the configurations used to create those raw files. Now I would like to look at them, so I'm going to open up a text editor. In this case, I'm using Notepad++. You can use whatever you like. And I'm going to open these two configuration files in the text editor. So if you're accustomed to looking at MZML files, you would probably realize pretty quickly that a lot of the information found in these configurations does not appear in the MZML file. So it's important to remember that a raw file contains piles of information above and beyond what appears in an MZML representation of it. So this is our effort to capture more of that information through Thermo Raw Meta Dump. Naturally, Thermo Raw Meta Dump is useful on instruments made by Thermo, but it is not useful on instruments made by Bruker or SciEx or Agilent. So you're not going to be able to use this tool there. We do not necessarily have tools that allow us to extract methods in the same way from those other experiment types. All right, so we're going to drop down through here. You can see that how the pump was configured, how the reverse phase shows up, all appears in here. Method duration, this is a 90-minute experiment. We have the spray voltage. We didn't actually use negative ion mode, so that one doesn't really come into play. And we see that we have these definitions of experiments. We have the start time. This runs from minute 5 to minute 90. It has a cycle time of approximately 3 seconds. And we see that it's going to collect a master scan. This is the MS scan. It's collecting it at 120,000 resolution. The mass range is normal, blah, 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 blah. And then we have filter states that are filters that are being applied to determine which precursor ions are allowable. In this case, because we're doing a top-down experiment, we are ruling out low charges and focusing our attention on charges 5 through 24. We are using dynamic exclusion. This is very similar to what you'd see in a bottom-up experiment. Its expire time is set to 180 seconds, so a particular mass to charge value will only be examined every 3 minutes. And we see that we have signal to noise criteria, etc. But what's not in here is a concrete list of what mass to charge values are interesting. We have precursor selection range that spans basically the entire range of the MS spectrum. We have the decision events and so on. And then we can collect a data-dependent MS scan acquisition that actually produces the tandem mass spectra of individual proteoforms. We have the collision energy and stuff like that. You might note that in the case of this raw file, the postdoc who collected this experiment did a really good job of trying to represent the instrument method. He has R120, R120 to reflect that both the MS scans and the MS scans were collected at a resolution of 120,000, and HCD20 here represents that the activation type was HCDN20. So if you are looking at data in the wild, we do not frequently see all of that material provided as part of the file name. So we need to look at the instrument methods to deduce that, if that is relevant to how we're going to do our data analysis. We see that we're collecting 8 microscans per proteoform, which means that the tandem mass spectra that come out of this may not be plentiful because the instrument is spending so long on each MSMS, but the individual tandem mass spectra should have relatively high quality. And as we drop down further, we see that we've hit the trailer on the file. So that's the complete description, and nowhere in this instrument configuration does it explicitly state the list of mass to charge values that are to be sampled. That is entirely data dependent. Neither is there an exclusion list here. It is possible for them to say, sample anything but ignore these particular M/Z values. We haven't done that in this case. Now that's all looking rather different than what we have on this other experiment. Both of these were collected on exactly the same Thermo Fusion instrument. The other was collected in March, and this one was collected in July of 2014. We used different energy levels. Here we see that we're using HCD 25% rather than 20%. But as we work our way down through the file, we'll see that there are probably more similarities than differences, because people tend to sort of fall in love with a particular set of configuration options. But this will certainly have some big changes to reflect that this is a targeted, or rather an inclusion list experiment rather than a shotgun one. We see that the former experiment was 90 minutes in duration. This one is 45 minutes instead. We see that experiment 1 is to run from minute 3 to minute 40, and that it's collecting a mass spectrum at 60,000 resolution rather than 120,000. The scan range is quite curtailed. It's a very narrow mass to charge range by comparison with what we saw before. And as we drop down further, we see that there are much tighter limitations on which charge states are to be considered. And finally, we get down to a targeted inclusion mass list. This is where the method specifies what is to be targeted in this experiment. We see that the mass to charge values are listed here, 581.8 and 634.6. And then we see that the instrument is told to sample whichever one is more intense. And then we have a scan event that defines how that tandem mass spectrum is to be collected. In this case, it's a tandem mass spectrum. We're not using something where we fragment multiple precursor M/Zs, just one. And we are defining a particular M/Z range that's already been listed up there. It's still HCD. Here we are using a collision energy of 25% rather than the 20% we had before. As we drop a little further, we see that there's a little bit more information provided down below. I'm going to just drop down to it. So here we see at lines 363, there's a second mass list table. So before we had 2 M/Z values that were lower than 700. Here we have 2 higher mass to charge values. So if you stopped at the first one, you might think, okay, there are just 2 targets for this inclusion experiment. But in fact, there are 4. The M/Zs we saw above, and 727.5 and 800.15. So a total of 4 targets, 4 inclusion mass to charge values that have been defined here. And again, we have a fair number of microscans being collected for these, so that the individual tandem mass spectra should be of pretty high quality. Typically, when people are doing inclusion experiments, they're doing them because the proteoform that they want to sample may not be the majority one in the sample. So frequently, you're trying to dig a signal up out of the grass to some extent when you do an inclusion experiment rather than a shotgun one. Okay, so we've finished this first step. We've read the method descriptions from the raw files using Thermo raw meta dump. Just because I used a particularly recent version of ProteoWizard does not mean that you are obligated to do the same. Thermo raw meta dump has been in ProteoWizard for several years now, so you should be able to find it in your install of ProteoWizard already. All right, so now we're going to start up Xtract itself. And I'm going to note that some of the default options seemed a little problematic for some of the data files I used it with. So I'm going to suggest 3 different things that we want to change within Xtract to possibly improve its performance in my data set. It is possible that you will have a different set of preferences for how to do it, so you should not treat these as canonical. These are just my opinion. And I'm pretty new at FreeStyle, I must say. So here's FreeStyle. This I think of as being sort of a successor to a development built on top of XCalibur. In the old days, you would have to do your deconvolution using Xtract within the XCalibur interface itself, and I think that FreeStyle makes it rather easier to do a lot of the operations that we would like to try. So I'm going to start by putting us in default options. This is something, I'm going to pull this away for just a moment. Default options appears on the Workspace Options tab. So if you've got Workspace Processing open instead, you won't see it there. So make sure you've got white Workspace Options pressed first, then Default Options. And within that, we have a huge number of parameters that we can consider. The only one I'm going to draw your attention to at the moment is Xtract options. We have things like fit factor and remainder thresholds and consider overlaps and so on, but the one I really want to draw your attention to is this very last value. You see that the signal to noise threshold is set to 3, and that's pretty reasonable, frankly. But some of the files I was working with in this experiment are pulling precursors from very low in signal. As a result, they don't stand up around among their neighbors very well, and I don't see all the spectra if I leave the signal to noise threshold where it is. So for this experiment, I'm going to set this to 1 and then click Save to force the software to save that as my default configuration. And that will be persistent. The next time I open FreeStyle, that default option should still be in place. Now the other options I want to change are only going to appear if I open a raw file. So in this case, I want to open the very first, the inclusion experiment that we mentioned a moment ago. So I can either create a new workspace or simply drag the raw file to it. You see that it has its preferred place to open, but I'm going to open the research directory on my computer, pop over to the directory for Helicobacter top-down, and then open the FreeStyle tutorial. And you can see that the inclusion experiment is right here. You can see the whole name in long form if I change to details. There you go. The inclusion experiment is right there. I'm now going to click Open, and that will cause it to open here within Thermo Scientific FreeStyle. Ta-da! Okay, and you can close the silly yellow notification bar. Right, so I wanted to point out that we do not currently see the options for Xtract up at the top. That's because I need to open Workspace Processing for that. So as I click Workspace Processing, I can click on Extract Deconvolution, and over here at the side, I get the options showing up. So if I pull that, I'm going to make that a little bit wider even. I want to point out that at the moment, the options are shown for Extract All. That's different than if I'm looking at an individual spectrum. Because if I'm looking at an individual spectrum, I clicked on spectrum 1, suddenly this changed over to the Extract Apply option. So you will want to keep in mind whether this reads Extract or Extract All, because one is applying to this individual spectrum and the other is applying to the entire raw file. So I'm going to click on the chromatogram again, and my Extract All option comes back. Okay, I want to note that, so down here we see that the output masses are currently set to M rather than MH+. This is very important because if you specify M, the deconvolution is going to try to produce neutral masses from charged fragments and charged precursors. So it's going to create a neutral mass for the ion, the neutral mass that corresponds to the ions. If I selected MH+, it would instead try to target the singly charged values rather than the not charged versions, which means that we have a proton difference between these two. It would be very handy if everyone in the field of top-down proteomics agreed on whether a deconvolution produces a neutral mass or produces a singly charged mass, because it's not frequently labeled in such a way that you know whether this tool expects a mass or a neutral mass or a charged mass. I'm leaving it default for now, so we'll just leave it as such. I'm doing protonated ions, not potassiated or sodiated ones. But let's get down to the options that actually produce a pretty big difference. You can see that Xtract is expecting to produce deconvolved masses that from things that are charged between +5 and +50. And that's quite a broad range, and the +50 is not really going to cause us a problem here. But I want to specify that when we're deconvoluting fragments of proteoforms, it's very frequently the case that the charge range needs to run all the way down to +1. Because if you break a fragment off of one that contains just one end of a very large proteoform, it may easily take on a single charge. So I would strongly suggest that when you're dealing with tandem mass spectra of proteoforms, that you allow for a charge range all the way down to 1. The other feature that will cost you an awful lot of sensitivity is this min-num detected charge option. This is a way to clean up the deconvolutions that result from Xtract, but it assumes that a given ion will appear in at least 3 different charge states within the mass spectrum or the tandem mass spectrum, and that is far too strict a criterion for tandem mass spectra of proteoforms, because very frequently we will have only one charge available for a particular fragment ion. So I need to change this to 1. All right, so I've now made changes in the default options for Xtract, and I've changed the charge range and the min-num detected charge for Xtract all. Let's go ahead and run the software. All right, now this is the Extract all is a new option as of version 1.8, and I have to say it's very, very handy because some of the files we want to deconvolute have dozens or even hundreds of precursor masses in them that we want to deconvolute. So if I had to do those one by one, I would be very, very frustrated, and I frustrate easily. But here I see that because it's extracting each of these scans, it has quite a lot of work to do. So let's take a quick moment to look at Task Manager. Task Manager, here we go. Is this software keeping my processor busy or not? What we see is that the CPU in my laptop, which has 4 different processor cores, is a little busy. It's not using all 4 cores at once. That would result in a 50% load, or all 8 threads at once. That would give it a 100% load. Instead, it's tooling along below about 30% load. I would, however, point out that something kind of unexpected is happening. In the background, I also see that the graphics processor unit on my CPU is busily firing away, also contributing to the deconvolution of these tandem mass spectra. On a computer that lacks some of the advanced instructions of modern processors, we can see that this deconvolution process takes quite a long time. I would note, for example, that I am not doing the data-dependent experiment through Xtract in this example, in large part because it takes dozens of minutes on my laptop to process. We also note that this is a fairly intensive operation. My CPU is already at 94 degrees Celsius. It's using rather a lot of power to do this, and the CPU responds by baking a bit. So it's fairly warm down there. So why does this take time? What's expensive about CPU time in this process of deconvolution? For this, I'm going to return to our little text summary. I'm going to just work my way through this. For deconvolution, we really mean quite a large set of different operations. So the term deconvolution has what programmers often call feature creep. That deconvolution used to mean one thing, and now it means a lot. So in the bottom-up world of proteomics, we don't typically deconvolute our spectra, which means that if we're trying to identify a +2 version of a peptide rather than a +3 version of a peptide, the fragments we predict to be present in the tandem mass spectrum differs, even though it's the same input sequence. The reason for this is in a +3 spectrum, we must consider the possibility that each fragment appears at either +1 charge or at +2 charge. So for every B ion, we predict a single charged version and a doubly charged version. In a +2 spectrum, we don't do that. We just predict that each fragment ion would be singly charged. So we're doing something very differently then when it comes to top-down proteomics, and that's because there's a very wide range of charges at which we would expect to see a given fragment ion. Okay, so let's walk through these 5 different roles that we anticipate for this deconvolution process. We have feature detection, which is to say that a proteoform does not typically appear in a single mass spectrum and then vanish thereafter. Instead, we typically see a rapid rise as that proteoform ion first appears in chromatography, and it lingers for perhaps 30 seconds, and then it goes away. So we have this sort of rapid rise and a slow fall off of these precursor features as we see them in the MS signals. So recognizing that a particular ion in one MS scan is the same ion showing up in the next 2 or 3 or 10 mass spectra is called feature detection. And frequently, feature detection includes something like integration of intensity to give us some sort of area measurement for that feature. Scan averaging is very useful when we have lots and lots of MS scans that represent the same precursor. So this is particularly necessary in experiments that use inclusion lists, because we fully expect that we're not going to produce a single tandem mass spectrum, but a whole, perhaps dozens of tandem mass spectra of the same precursor. So averaging those together gives us the opportunity to produce a much more accurate tandem mass spectrum, one that's much more sensitive for individual fragments than if we look at a single tandem mass spectrum absent the others. We also need to carry out charge state inference. We have precursor ions that appear in the MS scans, and we have an isotopomer envelope, which is to say we have a set of different isotopes that all belong to the same chemical composition, albeit at different isotopic enrichments. So if you have a monoisotope for a proteoform, all of its carbons are 12, all of its hydrogens are 1, all of its oxygens are 16, and so on. However, if a single carbon nucleus is a C13 rather than a C12, it's going to be spaced 1 mass unit higher. But on mass to charge values, the spacing of those isotopes will depend upon what is the charge on that ion. So if you see that the isotopes are separated by 0.2 rather than 1, you can say to yourself, 0.2 is the reciprocal of 5, which means that this is so 1 divided by 0.2 is 5, which tells us that this is a precursor that's charged 5. So this charge state inference is also something that we make part of this deconvolution process. As I mentioned before, the software is either going to output a neutral mass or a singly charged mass as its output. So charge reduction is necessary for this. If you see a fragment ion at +5, you're going to try to discern what its singly charged or neutral mass should be from that. And in some cases, you may have a fragment ion observed at multiple charge states. So if the same fragment is observed at +5 and +6 and +7, added together, those can produce a very strong signal in neutral mass or in singly charged mass, drawing from all 3 of those charge states, all of those overlapping at the same mass. And finally, de-isotoping. We have an isotopomer envelope, and we're not going to show all of those different isotopes in the output spectrum. We're going to de-isotope them, hopefully producing a single peak at the monoisotopic mass for that entire proteoform. So all 5 of those processes are taking place, and it's going to take a little bit of CPU time. As you can see, we are sitting at scan number 803 out of a little less than 1,000, if I remember correctly, for this raw file. So I'm going to now return to the Xtract user interface. Its dialog should be sitting on top of it. And we can see that our 40-minute experiment here is wrapping up nicely. 850, okay. One of the questions that I want to mention here is one that I find a little bedeviling as I try to work through inclusion experiments. If you recall, when we pulled out the instrument method using a Thermo raw metadump from this raw file, we saw two sets of two mass to charge values. And they had slightly different information, or slightly different configurations, I think, on how they were to be fragmented. So you might think to yourself, if this inclusion list included exactly 4 masses, surely what we have are a large collection of tandem mass spectra from exactly 4 masses. And I think I'm going to surprise you when I show you what actually resulted from this. And I'm going to wait for Xtract to finish here before I show you that little surprise. It's almost finished. It's oh so close. There you go. Perfect. All right. Now, before we look at the result of the experiment, I'm going to go into the MSN browser. It's just another one of these panels that appears over at the left. You remember that I said we had 4 different target mass to charge values. But look at this tree of precursors that resulted. I have a precursor of 582.1035, one of 634.8908, one of 727.45, another of 727.95, one of 800.1060, and 800.6592. You can see that the instrument has done something a little creative here. It had 4 precursor masses, but in the end, it split them up to 6 different precursor masses. So these 2 masses, both 727, were pretty close to the targeted mass to charge value specified of 727 point something. I can look back over at the method if you'd like to see that. Down here, we see that 800.1060 and 800.6592 are very, very close together. So our 2 high mass to charge value targets have each produced 2 different masses that have been recorded in tandem mass spectra. I want to specify as well that some of these are average spectra and some of these are individual spectra. So if I were to double click on this, I would see that I've got my spectrum showing up down here in the corner. Now I want to see that one all by itself. So I'm going to close the first spectrum that was opened just by default. And I want this chromatogram to go away. But if I click this X, I got to tell you it's going to close the entire raw file. So don't click the X. Instead, we can use the auto hide feature to make the chromatogram vanish. And now we have just our average spectrum for this mass, 582.1035. This is an average spectrum representing spectra all the way from scan 5 to scan 977. And you can see then that this is the set of M/Z values that correspond to it. That looks a little different than if we look at a single spectrum by itself. I'm going to go ahead and pull that one open also just by itself. This is an individual tandem mass spectrum collected for 727.4556. Its scan number is 787. And you can see that the other tandem mass spectrum that was collected from that same targeted mass to charge value includes scans all the way from number 308 up to 959. Does that look the same? It actually has quite a few similarities with the other, but in this case, the instrument thought that the spectrum for 727.45 looked different enough that it treated it as an individual, a singleton spectrum, not as an average spectrum. Now, the next step that I'm going to tell you to do may seem a little counterintuitive, given that we just did all this processing and we want to look at the result of it. But the result of it is not here. This is the result for the original raw file, and what we want to look at is what was produced by the deconvolution. So let's close this particular raw file. And now I'm going to come back to this directory. We see now that I'm going to sort just so the raw files all show up together. Before, we had these 3 raw files, because remember, I pre-processed the data-dependent one. And now we have an added raw file, and it didn't ask us what to name it. It simply called it the same raw file name with underscore extract on the end of it. This is our signal that this raw file is special. It is one that has already been through the deconvolution process. Now I'm going to maximize that, and I want to show you something a little surprising here. This raw file is not just changed in name very superficially, it's also changed dramatically in size. And something I'd like you to remember here is that deconvoluting a spectrum, generally speaking, is going to drastically reduce the number of points recorded. Generally speaking, broadly, we often find that people collect top-down tandem mass spectra in profile mode. And instead of representing the spectrum as profiles, we tend to represent the deconvolution in centroids instead. So we're giving a list of masses where before we had a sampling of the entire peak shape. The other thing is that a proteoform tandem mass spectrum is likely to be highly redundant compared to the tandem mass spectrum in deconvolved form. The reason for this is that a single mass in a deconvolved tandem mass spectrum represents multiple mass to charge values, not only because a given mass may appear in multiple charge states, but each of those multiple charge states appears in multiple isotopes. So we're really, really simplifying the spectra when we produce a deconvolution from it versus what it was to begin with. And thus the raw file size drops dramatically. Now, if you're really excited here about the prospect of opening this raw file in something from ProteoWizard or something from OpenMS or something from any of the other toolkits out there, I have some very bad news for you. These Xtract produced raw files are not really usable in anything but Thermo software at this point. I made the attempt to open one of these extracted raws within the ProSite PC software and didn't get very far. It's in a slightly different format than the Xtract that was embodied, that was integrated into the XCalibur software. So we can still open these in XCalibur, and we can still open it in FreeStyle, but we can't really open it in something like MS Convert, where we might want to send this file to another toolkit. Okay, so for now, I'm going to pull this extracted version of the raw file into FreeStyle. And again, it's given me a little notification here. I'm going to make that one go away. All right. So when we look at our MSN browser, again, we see that there are 6 distinct mass to charge values that were observed to meet the 4 precursor M over Zs that were on our inclusion list. And when we look at individual spectra, they look very different than they did in their original forms. So if I want to make this list go away, I'm going to turn on its auto hide. Now I can look at these precursors and ask what they look like. So the average spectrum that we produce from the first precursor has a whole smash of peaks down here at low mass and relatively few peaks as we move upwards. That one at a glance looks rather difficult to interpret. If I were looking for a sequence tag, for example, I think I would really struggle. Our precursor here has some amount of noise in it. I shouldn't call that noise. It has some amount of low intensity peaks. Some of those may be viable fragments. It may be possible in this case to interpret a short sequence from some of those. But if I look over at my research notes, I'm reminded that there was a particular spectrum I wanted to look at. I wanted to look at precursor 6348908. Is that the one we're just looking at? It is. How wonderful. Okay, so this is an average spectrum. This is another thing that's a little hard to convey, so I'll try to walk through this. The spectrum of masses that we are looking at here is labeled in mass to charge values, but in fact the charge that we've given it is 0. So this is not a mass to charge spectrum. This is a mass spectrum where these are deconvolved masses with a neutral mass reported. So don't allow that labeling to fool you into thinking it's something other than that. Okay, we wanted to look at a particular spectrum though. I was going to look at the 634 here. Well, it's okay. So we are looking at the fragments that are produced from a particular precursor. This is one of the proteins that this experiment was really trying to interrogate closely, to be able to do a match in the ProCyte software. And it was in fact something that we could match very successfully to a novel sequence for a histidine-rich protein found within Helicobacter acinonychus, which is a relative of Helicobacter pylori. They're pretty closely related species. Okay, so I'm not going to show you the identification process, nor am I going to show you in this video how to align these two sequences. That's something that we're going to do in the next video, which will be about ProCyte Lite, which is some free software for aligning sequences to an observed tandem mass spectrum. And I'm also going to show you how to do this in Clips MS, which is some Python software created in Joe Liu's group. So, Carter, what's his last name? I'm sorry, I don't mean to slight you. I'll make sure I have his name in mind when I show you his software in the next video. I do want to note, however, that I might want to be able to look at the at the mass list rather than looking at it in spectral form. And for that, I can pop up here to Reports and get a spectrum list. Here we have our list of these. You're probably accustomed to seeing masses sorted by the x-axis here, by the mass axis. But in this case, we see that the default is actually to sort the peaks by how tall they are. The tallest peak in this spectrum is at 6957, this one over here. It's a 100% relative intensity, and then we get down to one that's a third its height, about a quarter its height, about a quarter its height, and so on. So you can change that simply by clicking these headers, but remind yourself that these are masses, not mass to charge values. Okay, was there anything else I wanted to show you here? I don't think so, but I like to be able to look at this and see not just the data in graphical form, but also the data in tabular form. It is possible to copy out of here and paste to other applications. But for now, I'm going to close this raw file, and instead, I want to look at an individual tandem mass spectrum seen in the original DDA raw file that we produced from the same species, from acinonychus. So I'm not going to open the extracted version, I'm going to open the original right now. Okay, and it opens pretty quickly. I can make our chromatogram go away. Certainly I would like to be able to set defaults so it just always does that for me, but in this case I don't get that option. Now let us look at an individual spectrum from this. We're going to look at scan 924. Remember that because this is a DDA experiment, we don't get dozens and dozens of copies of the same tandem mass spectrum. So we're going to look at 924. Oh, it's already open. Right, so I happen to remember I need my chromatogram for this. Okay, so let me pull open the chromatogram. Here I see that I wanted to look at a particular scan number within this. Oh, I need to actually turn off its auto hide because I've been grouching about it and now I need the software to show it properly. And maybe if I'm very, very lucky, I will get the mouse in just the right spot and turn that off. Okay, auto hide is back. All right, so now I want to find scan 924. I remember it's a little bit after this peak. I can see my scan numbers appear right down here. I'm going to just use the left arrow and I'm holding it down. You know, I want to get a little closer because this is going to take a while. Click right in there. Okay, 928. left, 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 and now I'm there. Here is spectrum 924. I really like this spectrum. Now I can turn on my auto hide again, and I have just this tandem mass spectrum to look at. Look at this beauty. Okay, this is a mass to charge tandem mass spectrum. It has not been deconvolved because I opened the raw data as they initially appeared. Is this an individual tandem mass spectrum, or is this an average? In this case, I'm looking at scan 924. If I come over here, oh, I'm sorry, I'm not looking at the correct part of the MSN tree. See that my precursor is actually 1205.41. I'm going to pull down here to 1205. Here's 1205.41 right there. Great. This is an individual tandem mass spectrum. I want to do a deconvolution, but in this case, I only want to analyze this individual tandem mass spectrum. So when I click on Workspace Processing and say I want to see Xtract deconvolution, I see that I have all of the same options that I had before. And because I haven't closed FreeStyle in the meanwhile, it remembers I want to see charges down to +1, and I want to see even the fragments that appear at only a single charge. But you see that right now this is set to show Xtract all, which means I need to remind it by clicking that I'm interested in this individual tandem mass spectrum. I see that that now reads apply, so I can click this single button and it will show me the result of just that individual tandem mass spectrum's deconvolution. And it doesn't take a huge amount of time because it's doing a single spectrum, not all 1,000 of the spectra that were collected here. So my tandem mass spectrum originally appears up here. Its deconvolved spectrum appears here. And in this case, the spectrum list is shown automatically. I automatically get my list of precursors. My list of fragments. But I want to note that in this case, the relative abundance is not the sort order. Now the monoisotopic mass is labeled monoisotopic mass instead of M/Z, and it's sorted by that list. So that's very helpful. This is more what most people, I think, would expect when doing a tandem mass spectrum. But be aware that the spectrum list that you get from the Xtract all raw file is different in its labeling and in its ordering than what we see in this case. I click back over to my little itinerary for today. I remind myself that this scan, number 924, was identified with this precursor mass to charge value to a major protein known to bind metal in H acetinitrous. I used the ProCyte PC software against an extracted peak list produced within an older version of XCalibur, not this particular FreeStyle. I want to show you something really important, though, when we try to look at that same spectrum in its deconvolved form in the Xtract all version of that experiment. So I'm going to return to FreeStyle. I'm going to close this raw file. And now I'm going to open the version of the raw file that we produced through Xtract all. Once again, we see that the file size dropped precipitously from 120 megabytes down to 3.5 megabytes. So again, we save a lot of space by storing the extracted version of the spectra rather than the full form. All right. So I'm going to reopen this extract version of the file. Remember from our little notes that we wanted to look at scan 924 precursor 1205.41. When we go to the chromatogram, we try to click on it directly, but we're not quite there. We move left to 928, to 927, to 926, 925, 924. When we look at the same scan number, suddenly our deconvolution looks awful. I just have 2 peaks here. How did that happen? Well, the answer is that something has changed. Before, scan 924 corresponded to a precursor M/Z of 1205.41. When we look at the precursor mass, though, we see that the precursor mass to charge value has changed. It's 1483.43. So even though this is the extracted version, or the extract all version of this raw file, the scan numbers are no longer staying in the same place. Scan 924 has moved to represent something else. So I want to find that spectrum. I'm going to use the MSN browser to go there instead. I'm going to scroll up to 1205 because that was the mass that we had. Do you remember 1205.41? Do we have multiple spectra? No, we just have a single spectrum. So I'm going to click on this. We see that our spectrum number has changed from 924 to 917. Now, why this is happening, I don't have a very concrete answer. I think it relates to the fact that in some cases, these tandem mass spectra represent multiple scans drawn from the original spectrum. Or perhaps the software is shy about showing us tandem mass spectra that have been deconvolved that produced 0 peaks. It may be that those have been stripped out. Something like that has happened so that what was originally scan 924 has become scan 917 instead. And once again, we have an interesting tandem mass spectrum in deconvolved form with lots of good peaks. If we pull open the spectrum list, we see that the software once again sorts it by relative intensity and shows M over Z values rather than masses. If we sort by the M over Z column, 321 is once again the first peak observed in this deconvolved mass spectrum. So keep that in mind. The spectrum, you might assume that running Xtract all will preserve scan numbers. That is not true. In some cases, we find that scan numbers change between the original raw file and the extract all version instead. Please be aware of that. Remember that deconvolution now means a whole host of different features. It's not just trying to separate out which signals in the MS scan relate to each precursor. There are a whole bunch of things going on about charge state inference and charge state reduction, scan averaging, and de-isotoping. I hope that you find these two raw files are useful in understanding just how differently two raw files from the same metal-enriched proteins from HS and NHS can look. Certainly a data-dependent acquisition experiment and an inclusion list acquisition experiment will differ quite significantly in how many spectra are available for each precursor, and it will change also the kind of deconvolution result you want to look at, one that is built from the average spectrum versus one that's built from an individual tandem mass spectrum. But I hope that you found this useful as a little guide to how to use FreeStyle in top-down proteomics. In our next presentation, we plan to show you how to annotate these deconvolved spectra to show in a fragment map and in an observed in the spectrum itself which ions have been labeled for a given sequence. But you'll have to wait till that next video is presented and shot. In the meanwhile, I hope you find this and other videos from our top-down series useful. And thank you very much to the EPIC XS program for funding all of these training videos. I really appreciate that. And for giving me a job, heck. Thank you also to the laboratory of Julia Shimoruk at Pasteur for having produced these raw data. Igor Vorontsov, who's now a staff scientist in industry, did some really fine development work in the exploration of different methods for this experiment. So thank you also to him. And with that, I will call it quits.